Hi, my name is Phidias Diamandis, and I'm a neuropathologist at the University Health Network. And I want to begin by saying thank you to the organizing committee for allowing us to present some of the work we've been doing on using unsupervised deep learning workflows to resolve intra and inter-tumor patient heterogeneity. So let's begin by just giving a quick overview of what kind of conventional supervised deep learning approaches look like for digital pathology. These tend to try to collect a large number of annotated images, such as shown here, label them with tags of interest, and then use these large image data sets to train a neural network to find specific spatial patterns in these images that are of diagnostic value for future classification tasks. This process is laborious, but once they're trained, they work very, very well at analyzing future images pixel by pixel, finding those specific feature representations, passing them on to deeper and deeper layers where the feature representations become more complex. And then finally, in the final layer, the most complex and informative feature representations are used to make classifications decisions. And this works extremely well. This is an example of one of our early experiments where we took this sample of brain biopsy from a patient with a glioma, and you can see the tissue is quite fragmented. And while running it through a neural network, we can show exactly in which fragments the tumor is located, as shown here in the pseudocolor map with the brown color. And this is extremely accurate when you can compare it to immunohistochemical stain for IDH1, which definitively labels the fragments that have the tumor. You see that there's almost a one-to-one correspondence with where this neural network detected the tumor. And as you zoom in, you see that the neural network can do a very good job of even detecting small collections of infiltrating tumor cells through the normal brain tissue shown here in pink. And this is what we've come to expect from neural networks, and it's because of this property that they're going to really transform digital pathology and computational pathology in the years to come. Now, we've started applying these to more real-world examples, and in these specific cases, we've seen a number of challenges that really become the main focus of our lab to try to overcome the challenges of applying these in real-world settings. Some of the difficulties, of course, is when these neural networks encounter relatively uncommon tumor types that may have not been incorporated into the training data set as yet. An example of this from one of our recent studies was a hemangioblastoma, a relatively uncommon tumor type that we see only occasionally. And because it wasn't in our training set, when it's encountered, the computer obviously has to try to guess and classify it into one of the known tissue types that it's been trained on. And in these particular cases, there's no chance for that it's going to get this case correct. Another problem is uncommon subtypes of common tumors. So gliomas are relatively common tumor types that we have in all of our classifiers, but there are in fact some very rare forms of glioma, such as gliosarcoma, that are only occasionally seen, again, maybe once or twice a year. And in these cases, you can see here, the tumor has two components. It has a glial component that labels with GFAP. In this case, it's only a minor component of this tumor, while the majority of the lesion has a more sarcomatoid pattern, which is labeling here with reticulin. And you can see because of this heterogeneity, the classifier got confused with the kind of sarcomatoid component and called it a meningioma in this case, which is obviously not the right diagnosis. And of course, as we kind of wrap our head around these problems, we see more and more challenges that could potentially occur. What happens if for these rare tumor types that we're training our neural networks on inherently have a lot of incorrect ground truths because of their relatively uncommon nature? What happens when new diseases arise that may not be in training data sets, as we saw this year with COVID-19? And what happens when the same diseases change due to changes in practice, such as immunotherapy or other treatment modalities that may change the morphology prior to biopsy? For example, I see a lot of recurrent tumors, and in these cases, they look remarkably different than the non-treated primary tumors that we're typically accustomed to seeing. So we want to try to address these challenges to make these networks more generalizable to everyday use. And the way that we're approaching many of these problems in our lab is using unsupervised workflows. And hopefully I'll try to explain to you what these are and how we're using them. So here is an example of the traditional workflow that we're accustomed to. We take these large digitized images, break them down into these manageable image tiles of 1024 by 1024 pixels, and then run them through these convolutional neural networks that are trained to recognize specific classes of interest. And then we get these nice pseudo-color maps with a classification score that allows us to understand where and what things are on the slide. Now, one of the things that we're interested in is trying to understand how these decisions are made, because if we can understand them, then we can leverage them in more and more applications. So we became interested in this final layer as a readout for the most complex features that a computer can generate, and trying to understand if they resemble the same features that we teach our residents to make intelligent decisions when working up cases. So to do this, we extract this 512 dimensional vector that's found in the VGG19 network and go feature by feature to try to understand if any of those high order complex features that are found in this pentultimate layer resemble what we look for under the microscope. Here's an example of the feature representation 215, and you can see that when we superimpose this specific feature onto this image of a metastatic colorectal cancer, it localizes to the kind of fibrotic core of this papillary structure, suggesting that this is a kind of fibrosis correlate which a human would interpret. Other features of this 512 dimensional feature vector, such as feature 165 and feature 66, as you can see, label different aspects of the image. They label this epithelial lining, and so we've now annotated these as epithelial correlates that the computer has generated. Here's another two features that we found, diagnostic value for metastatic tumors. These are luminal correlates. As you can see, the activation occurs in this empty space within these gland-like papillary structures, and you can imagine that when it comes time to classify it, the computer, like the human, uses features to provide a specific class. Now, of course, humans are very good at translating these kind of fundamental features into novel environments, and that's what makes us very good diagnostic clinicians. So we asked, can we use these same principles and extract features from this brain tumor educated neural network and apply them to different novel scenarios? So the system is pretty much similar to what we've seen already. We take an image, break it down into these image tiles, and then run it through the neural network. But instead of trying to extract a classification, we instead use these features to cluster individual tiles and cases based on the similarities of these human-like constructs. And so we're trying to recreate some of these decisions, but in a kind of cluster-based, unsupervised approach. Now, why do we want to do this? Is that once we're able to cluster things based on similarities, we can redeploy these brain tumor educated neural networks to different novel environments. Here's an example shown here. This is a renal cell carcinoma specimen from the TCGA network. We apply our brain tumor educated neural network to cluster and subgroup the individual tiles that are derived from these images, and you can see that there are specific clusters that arise. When we cross-reference these specific clusters back to the digitized slide, we see a non-random distribution of clusters within the spatial coordinates of the slide. And when we examine these under the microscope, we see that in fact this system is able to in fact find unique patterns in tissue types that it's never seen before. And this is of course very, very important because it's scalable and allows us to use these in a very generalizable way. You can see here we have identified in the yellow region the clear cell carcinoma, but also identified very unique patterns that would never have been used in our initial brain tumor educated neural network, including normal renal parenchyma near the tumor that has undergone atrophy and fibrosis, and so we have a very generalizable solution for digital pathology. Here's another example of how we use these to identify intra-patient heterogeneity. So this is a slide from the TCGA network of a uterine carcinoma. You see the myometrium, the endometrial tissue in pink, and then the carcinoma is the more cellular purple area of the slide. We automate the tiling of this image and then cluster the specific 516 micrometer square tiles to see which ones should be grouped together. We color code the map, and then we can see that in fact it separated out the orange endometrial tissue from the carcinoma that has been labeled with the lime and red and green cluster. Now, interestingly, when we look at these individual clusters, we see that in fact it was able to subclassify different parts of the tumors with different morphology. So you see in the lime cluster, we see these representative tiles showing a gland-forming component, whereas in the red cluster, we see that the tiles largely form this kind of sheeting, poorly differentiated component. And so we now have a very nice system to measure intrapatient heterogeneity in an automated way. So we think that these are going to be the types of solutions that will help us overcome these rare tumor types that are otherwise very hard to train manually, such as in gliosarcoma. We're going to be able to really measure the heterogeneity and try to classify each of these subregions to find the most appropriate diagnosis. Now, what about new cases, or can we automate subclassification of new diseases or new patterns of diseases that may have not previously been encountered? So to do this, we wanted to use the renal cell carcinomas in the TCGA network as a proof of concept, namely the papillary and clear cell group, because these look very different to the pathologist and have very different outcomes for the patient. So that we thought that would be a good proof of concept to see if an unsupervised approach could recreate this subclassification scheme of renal cell carcinomas. Interestingly, clear cell carcinomas are also graded using this ISUP grading scheme from grades 1 to 4 based on their nuclear and cellular morphology. So as you go from grade 1, 2, and 3, the nuclear size increases and there are prominent nucleoli. And in grade 4, you see this very atypical sarcomatoid pattern that represents the most aggressive forms of renal cell carcinoma. So we wanted to see if we could also create this classification scheme that obviously required many experts to come to consensus on how these should be graded and their prognostic significance. So lo and behold, we took the appropriate cases from the TCGA network for both papillary and clear cell carcinoma, asked the computer to cluster them as it see fit, and you can see here the computer decided to generate two major classes shown here in red and green, and you can see by the color of this TCGA class that these major subgroups proposed by the computer are largely driven by the TCGA class which they arise. We have the green cluster primarily composed of these papillary tumors, and then we have the orange cluster primarily composed of the clear cell carcinoma subgroups. So in fact, we're able to now recreate some of these expert type classification schemes in a very automated way without any training at all. So we wanted to see how exactly this works. So we looked at the feature representations enriched in these specific subgroups. One of the features that was overrepresented in the clear cell carcinoma subgroup was feature 219. You can see here when we find images that are activated for this feature, we see that in fact you have a clear cell morphology as would expected, and when you deplete feature 219 and look for images or cases that had a low feature 219 activation, you see that the cells have a more eosinophilic cytoplasmic pattern that is not very typical for a clear cell carcinoma. So this is kind of worrisome of a potential misclassification or a rare subtype of renal cell carcinoma. And this, of course, is transferable, so you can use this clear cell correlate in the papillary renal cell carcinoma TCGA cohort, and what you see here is you get this papillary looking tumor that have very clear cytoplasm faithful to this feature representation, and this in fact represents a clear cell variant of the papillary renal cell carcinoma that is now recognized to be a distinct entity with a very good outcome. So again, we are able to use these tools without any training and find very sophisticated subgroups in these large scale populations. So finally, we wanted to see if we could recreate the ISUP grading scheme using this same principle. Can we cluster the clear cell carcinoma cases and see if in fact the clusters correlate with either survival or the nuclear features that we're accustomed to. So again, to do this, we focused exclusively on the clear cell carcinomas of the TCGA network. We clustered them based on their feature representations. Here are the 512 features, and here are the 3 clusters that were proposed. And then we wanted to see if these had any clinical correlations. of significance. And here it is. We see that, in fact, they have remarkably different clinical outcomes, which was both surprising and exciting for us. So we wanted to see how these patterns were driven, if there was any kind of correlation to the expert knowledge that we now know based on these consensus guidelines. So we again used bioinformatic tools to identify the feature representations that were enriched in the superior and poor prognosis group. And so we can look at those features individually to see how they correlate using these feature activation maps that we've seen. So two features of interest was 442 and 499. These were interesting because we could understand what these features were in fact correlating with. So you can see here 499, when you find images that have a high 499 activation, you see that they're activated only in the tumor cells and not the stroma of the tumor. And you can see here very ugly nuclei with prominent nucleoli even seen at this magnification, whereas clear cell carcinomas without this feature 499 had very small nucleoli that are barely visible at this magnification. Similarly, 442, you can see it's one of the kind of most representative features enriched in this poor prognosis group. And you can see the histology here has a very sarcomatoid spindle-like arrangement that correlates with that ISUP grade 4 picture that we saw in the previous slide. Again, when you deplete this feature and you look for clear cell carcinomas that had a low feature 442, you see that this feature is completely depleted. Interestingly, these features alone are prognostic in the TCGA cohort in and of themselves, really highlighting exactly how we can start to recreate and identify some of these features purely by computation. We can again look at the reverse, look for good prognostic deep learning feature representations. Here are two that we thought were interesting. Here's feature 263, and what you notice is that you get these eosinophilic rich cells with these large cystic spaces in many of the images that registered a high feature 263 value. And you can see from the feature activation map, it is indeed not the tumor cells, but the actual cystic spaces that are driving this high value. Interestingly, 254 was another feature that was enriched in the good prognostic group, but peculiarly when you look at the histological pattern of this lesion, you see that in fact the cells are very eosinophilic rather than the clear cell morphology that you would expect in the clear cell carcinoma cohort. So we thought that was very peculiar, but again, just to see if in fact these are independent prognostic factors, you can see that the clear cell carcinomas that have a high feature 263 and feature 254 on their own, again, show a good prognostic outcome, again, validating that these are, a very, very kind of quantitative biomarker tools that we can now apply to our digital pathology workflows. So to really validate that this was in fact what was going on, we reason that many of these kind of eosinophilic rich tumors that were found in the good prognostic group were in fact misclassifications perhaps by the TCGA network. So we partnered up with Trevor and his team to look at the mRNA expression profiles as an unbiased approach to how these tumors ought to be classified based on their molecular data. And we got these three major groups. We have this green group, which is the clear cell carcinomas. We have this blue group, which is the papillary renal cell carcinomas. And then you have these chromophobes. We took these 9 outlier cases that had a superior prognosis and tried to see where their mRNA profiles were most similar to. What you see is that in fact 5 of the 9 cases were found in this chromophobe group, whereas 2 of them correlated with the mRNA profile of your typical clear cell carcinoma. And 2 were outliers, and when we showed pathologists, these looked like oncocytic tumors that are benign renal cell tumors. So hopefully that gives you a nice kind of overview of how unsupervised tools can be used to not only classify but organize tumors in intelligible ways without the need for kind of laborious continuous training with more and more training data. I want to thank Kevin and Adil for their hard work in this, who generated the majority of the data that I presented today, the entire team for really helping us analyze these in intelligible ways, Trevor and his team, Sangeeta and Andrew Evans for their input in this project, our funding sources, and I encourage you to kind of follow our progress and other lectures on our YouTube channel, NeuroPsyQ. Thank you very much.